Welcome, 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 friends. Welcome, wagon.
Blood. 10 pints of life force, coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving. Our red blood cells carry oxygen and nutrients. Our platelets stop bleeding and help to heal our wounds. Our white cells protect us against infection. And the yellow liquid plasma in which they are suspended carries an array of proteins that regulate bleeding and clotting.
Okay, so that was just a little intro of the components of blood. I like that image because it kind of shows you the cells. Okay, so I'm going to do something a little bit different today. I am going to lecture over blood components, and typically this would be a 2-day lecture in class, but normally like we would do a lecture and it's a short lecture, and then we would do something, you know, worksheet with it, and then the next day we do the other half, and then we do a worksheet with it. So rather than do this over like today and Monday, since our hours are Thursday and Friday basically, right? This hour and next hour. I'm just going to hit both parts of the lecture all at once, and then you can do follow-up along with it later, which actually we're going to do something different than the worksheets I typically do. So I created a notebook that goes with my lecture.
Yeah.
So you can fill the notebook out. If you want to split your screen, you can fill it out while I'm lecturing. Or you can do it as a reflective notebook after the fact. You can come back and see what information you can recall, and you can fill it out in later. And this lecture has already been pre-recorded, so if you wanted to fill it out and then use video to see if you missed anything, you could do that as well. We have a 3 levels reading guide. It's actually not on the schedule till Friday, but you might find that it's not on the schedule till Monday, but you might find that you have some spare time. Today, like if you fill out the lecture while we're lecturing, you're going to have some spare time, in which case maybe you do the 3 levels reading guide, which sets you up for next week. So it's up to you. The notebook's what we're turning in today, so you can either do it now or do it after. And then the reading guide is actually not on the schedule till Monday, but you might want to fit it in today and get it out of the way. All right. So blood components is fairly easy. We're going to have a quiz next Thursday. Today, you're going to get some descriptions. You're going to get some examples. Those are the kind of things I'll ask you on a quiz. You have intermission questions along the way. You can expect those are the kind of information that I'm going to ask you, on a test. So pay close attention to those as you study. All right. Does anybody have any questions, concerns before we get started? Monday, we did a pre-test. You might remind. So here we go. Let's get started on our blood. I love this part. I love the blood. I love the cells. I love the immune system. Those are part of my favorites. So when looking at blood, you have, we want to be able to talk about the 2 components, the liquid portion here, the formed elements here. And we want to talk about what the function is for every one of these cells. So we can see 1, 2, 3, 4, 5, 6, 7. That's all 1. We want to be able to talk about the function for each of those. So when you look at blood, it's like if you spun it in a centrifuge, it's going to separate out. You got this heavy piece separating out to the bottom, and the lighter portion is separating at the top. If you've seen a centrifuge, it goes super, super fast. Okay, so the plasma is the liquid part. This yellow piece up here, about 55%, yes, you do want to know the percentages. About 55% of your blood is made of plasma, and the majority of that is water, okay? So we're going to talk about what's in the plasma. Remember, connective tissues are kind of categorized according to their matrix, the material outside of the cells themselves. So this is our only liquid connective tissue. And the matrix, i.e., the matrix is the non-living portion. And then the cells, the cellular components, white cells, red cells, platelets that clot your blood, those are the formed elements, right? So blood is either liquid or it's a solid component, basically. Okay, we can see the formed elements. If the liquid makes up 55, the formed elements make approximately 55. The majority is red blood cells, and there's just a little bit that makes up the white cells and the platelets. This is called the Buffy coat, this little section in between. So when they spin your blood, they separate it out by weight. The plasma cells at the top. So the living component is the formed elements. So plasma, the liquid piece, we can see over here what it's composed of, mostly water and also blood proteins. So we're going to talk about the types of proteins we might find in them. Fibrinogen has to do with blood clotting. Albumin has to do with like your water levels in the blood itself. And then we want to know about those proteins. Hang on. So typically considered, so this is description, you'll see it on the quiz, right, in descriptions. Straw colored, makes up about 55% of the blood. We saw that in the centrifuge tube here, majority water. Nutrients, so this is the part that is carrying like the food supply to your cells and carrying away the waste products from your cells. So its whole job is transportation, right? So things like glucose that we use for energy, lipids, ions such as sodium and calcium, which we know is big for muscle contraction and nerve impulses, bicarbonates, which this plays a big role in your blood pH, different gases, oxygen, CO2, hormones. Hormones are part of your endocrine system. They're released from a gland. They travel through the bloodstream and they talk to other cells to cause a response. Similar to neurotransmitters, but neurotransmitters run through neurons and hormones run through blood. And they're going to take away the waste products from other cells. Okay. Oh, I might have that turned off. Miss Hefty, do I have that option turned off? It's in my settings. Okay, so antibodies that, blood proteins that I'll talk about here, the antibodies, the clotting factors, and albumin. So plasma proteins are not taken up by cells themselves. They will stay in the blood. Albumin, this one here, is important for maintaining osmotic pressure. Basically, that's like the water level of your blood, and that plays a role in the pressure of your blood. Antibodies, play a role in fighting against infections. So I got my bruise from my COVID vaccine. My body is now trying to recognize those proteins, and it's going to make antibodies against it. Clotting factors, hopefully you can recognize that has to do with, if you get a cut, you're going to start bleeding, and you need clotting factors to help close that cut. So those are three common plasma proteins. They're in the plasma itself. They're not in the cells themselves. The formed elements are red cells, white cells, and platelets. So I want you to know the scientific name. This will be on your quiz, erythrocytes. Remember, cyte means cell, erythro, red. So literally, it's red cells. Leukocytes are white cells. white cells, literally white blood cells, and thrombocytes are platelets. Okay, so thrombocytes are referring to your platelets. The red cells, and if you want to shorthand RBCs, that's fine. I know what you're talking about. Description, this you'll see on the quiz, flat, biconcave, meaning that it's curved inward on both sides. Anucleate, a not or without, nucleus. So we find structure meets function, right? So this cell is created to have a high surface area, like folding in the brain, increased surface area. Take away the nucleus so you have more space to carry hemoglobin and to carry oxygen. And that's the job of the red blood cell. So because it doesn't have a nucleus, it can't replicate itself. It can't go through mitosis. We know mitosis, remember, is the division of the nucleus. So if you don't have a nucleus, you can't divide it. So you can't make more red cells. They have a lifespan of only 120 days. After that, they're going to be decomposed in your spleen and in your liver. We will make more red cells in our bone marrow. So that's how they get replaced. So we have phagocytic cells. Phagocytic cells are eating cells. They gobble up old cells. And they'll be taken out through liver and spleen. Okay. Interesting fact. Now, I would not quiz you on this number, but you make 2.5 million red blood cells every second. That's kind of an amazing fact, I think. Okay, so those are red cells. Here we're going to look at that surface area, how folding increases the surface area, which one of its jobs is gas exchange, right? So this gives us more surface, so more gas exchange can occur. This is the second largest component of our blood, 45%. It doesn't have a mitochondria. So another important reason, or another important factor in its structure function, it's carrying oxygen. Mitochondria, remember, the energy part of our cell is using oxygen to make energy. So you don't want a cell that's job is to carry oxygen to use its own oxygen, right? So this is part of the cell differentiation for red cells, and it allows it to carry more oxygen. That's the whole purpose. So the large surface area makes it so you have greater room for gas exchange. We're exchanging oxygen and CO2. We talk more about that in the respiratory system. So we have red cells. These are your red cells. Oxygen molecules inside here, and they're bonding to hemoglobin. So we want to know about hemoglobin. Heme means blood. Globin is referring to the fact that it's a globular protein. It's a clump, a cluster. So you have about 250 million molecules per cell. Now the number is important because that is going to talk about how much oxygen your cells can carry. Each one of those can carry 4 oxygen molecules. So a hemoglobin molecule has 4 binding sites, 4 hemi groups. Iron allows it to carry oxygen. So if you have 250 million molecules of hemoglobin, each carrying 4, that means you can carry a billion oxygen molecules. So I think you do see that number on the test, not the quiz, but the test. Okay, so real quick, what kind of things am I going to ask you? Here's your intermission. So if you have your slide set open, you have a quiz yourself, your notebook. Open up your notebook. You have a quiz yourself. I'm going to give you the question. Miss Hefty, if you're in my drive, you can open up their assignment in Google Classroom. You know how we can work on it. And you can kind of flip through and see that everybody's working while I'm doing this. Okay, so what are the 2 categories of blood components? Everybody's going to write down these answers. This is like if we were in class, we would go over these together and then I'd call on you for answers. Miss Hefty's looking to see that everybody's putting answers down. She's just going to go scroll, scroll, scroll. What % of blood is plasma? These are all testable, quizzable pieces of information. You have that notebook that I created. It's purple. You have a slide that says quiz yourself. So you can scroll right down to that quiz yourself slide. What is the main function of plasma? What are some functions of plasma proteins? What percent of blood volume is red blood cells? What is the scientific term for red blood cells? What is structurally unique about erythrocytes? What is the main function of erythrocytes? How many oxygen molecules can one red blood cell carry? And what is the lifespan of erythrocytes? I think that's my last question. Nope, one more. What are the other two formed elements? So I'm going to give you a handful of minutes to answer those. So when we go back to in-person, we'll have shorter activities where you get a little 5-minute at the beginning, then a short lecture, and then another 5-minute at the end. So that this would kind of be the end of one of those, even though it's not even 45 minutes yet, but this would be more of a feel for how that hour would go, but we're going to go double today because we have a longer class period. You don't want to wait for me to answer them for you. A, I might call on you and then you'll be stumped because you didn't think about it in advance. And B, you'll miss the opportunity to quiz yourself. Okay, leuk meaning white, site cell. So leukocytes, and here's examples of 5 leukocytes at the bottom. They make up less than 1% of our blood. Normally we would do a microscope lab, we'd look at each of these under the microscope and you'd try to identify them. They are the only complete cells in your blood, meaning they have a nucleus. They have all their organelles. We learned that the red blood cells don't have a nucleus. We haven't learned yet, but thrombocytes aren't even complete cells, like they're fragments of cells. So the leukocyte is the only one that has all of its pieces and parts. It's essential in the immune response, which is chapter 12. So we'll be going circulatory system next and then into the immune system. Also found in the lymphatic system, your lymph nodes, right, under your ears, kind of in your neck. You have them in several places. We'll talk about them later. The spleen, wrong side, and in other body tissues. So these white cells, we'll talk about them. This is what Alex was telling us, the 2 categories of white cells. Cells are either granulocytes, meaning they're filled with granules, that's how their name comes to be, or they're agranulocytes, a, not or without, so they are without granules. So when I have granulocytes equal, they're filled with granules, agranulocytes equal, they are not filled with granulocytes. Notice the endings of these words. All of the granulocytes end in fill, P-H-I-L, fill. So I say they are filled with granules, right? Little speckles inside. So each of them have different nuclear structures. That's how we'll identify one from the other. When you're looking at them, you want to look at the nucleus. And they all have different jobs, okay? The agranulocytes, notice their ending is different. So that helps you categorize them. On the quiz, I'll ask you which ones are the granulocytes, which one are the agranulocytes. Just look at the ending and it'll help you out, okay? Lymphocytes and monocytes. So these red empty circles, and they're not really empty, it's just how the light is shining on them. Remember, they're biconcaved red cells, so they're thinner in the middle. So you're seeing right through them, that's all. So the smaller circles are your red blood cells. The bigger circles are your white blood cells. That's how you know the difference, little, big. Usually these are pink in pictures, and sometimes like on our microscopes, they're so light colored you barely see them. And then these are purple because they use different stains to help you see them, right? Notice the inside is dark purple. The inside is the nucleus. So this nucleus almost fills up this entire cell. Kind of looks like an eyeball. This is your lymphocyte. Whereas you have this giant kidney bean inside of a monocyte. The basophil, I can hardly see its figure 8 shaped nucleus, it looks like a puffball full of granules. So when I look at this under the microscope, that's what I see is just a whole bunch of purple circles. Eosinophils, these typically are described as like looking like the old school phone. Oh, I bought my mom an old school phone because she doesn't answer her cell phone. I gotta plug that in. So it looks like that old school phone, or like headphones, like Claudia's wearing. It looks like her headphones. And you can see how these have lots of granules in them, and then you can see these have less. And then the neutrophils, numerous neutrophils have numerous lobes, like they all start with nu, right? So it has a bunch of lobes inside, and that's how we'll identify one from the other. So neutrophils, we'll start with them because they're the most numerous. You learned that yesterday in your blood cell lab. So they are named according to the stain they use. So they use a neutral stain. You have an acidic, a basic, and a neutral. They use a neutral stain with these white cells, and that's how they got their name, neutrophil. They are the most numerous, most abundant cells of the white cells. Red cells are the most abundant, right? They are active phagocytes, meaning they engulf and eat. Remember we said you get rid of red cells by phagocytic cells. They're engulfing and eating. So they play a huge role in your immune system. They're multi-lobed. Talking about the nucleus, you can see this nucleus has 3 lobes, so it kind of gets divided into 3. It can be anywhere from 2 to 5. I think sometimes even 7. So when you have an infection, these are the cells that you're actually seeing in that festering of the infection. So like if you have a wound in the pus, that whiteness, that's these neutrophils. So these are common in acute infections, short onset infections.
Neutrophils are white blood cells that hunt and...
Am I sharing my sound? Did you hear anything? Oh, good. Okay. So this is the neutrophil here. These are red cells, and you're going to watch this guy hunt down a bacterium, and then it's going to engulf the bacterium. So you're going to get an idea of that phagocytosis.
Kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
And now the bacterium's inside of the white cell. Okay, so that gives you an idea. That gives you an idea of what that looks like. Okay, eosinophils. So they are stained with a stain called eosin, and that's how they get their names. They're bilobed. So see here, this one here looks like those headphones, right? Headphones or old-fashioned telephones, but I think they look like earmuffs also. So that helps us identify those. They have large coarse granules. So I can see these a little bit more prominent than I did in the neutrophil. They kill parasitic worms and increase during allergy attacks. So many of you have allergies. They can be brought on by eosin, eosinophils. Some of you have asthma that's brought on. You see that in a commercial, right? It's brought on by an like an increase in number of eosinophils. So if you have a parasitic infection, these numbers will increase. So when you go to the doctor's office and they do the blood draw, they look and see which type of white cell has an increase in numbers, and that helps them identify what type of infection you might have, which allows them then to maybe prescribe a general antibiotic that would attack a parasitic infection or a bacterial infection. Basophils, our last one, look at the big purple puffball. Stains dark blue with a basic stain. That's why it's called a basophil. The granules release histamine. Histamine is an anti-inflammatory. No, histamine is sorry, I was about to say something in addition. Histamine is an inflammatory. You take an antihistamine, right? When you have a cold or allergies kicking up, you take an antihistamine to get rid of the inflammation, which lets you know histamine causes that inflammation, right? It causes blood cells to dilate. So histamine and serotonin, and those are both vasodilators. Blood vessels will get bigger. Mediate hypersensitivity reaction. This is allergies. You're overreacting. You're being overly sensitive. So that cat enzyme from your cat's fur, it's not going to kill you, but your body overreacts and thinks that it's an invader, right? So you have an allergy against that. So these basophils are going to be active during an allergic reaction. They are part of your inflammatory response. These are the least common, so they have the lowest number in your blood. Agranulocytes, A not or without. Notice how clear the rim of this is, right? So the lymphocyte, its nucleus is almost as big as the cell. It's filled with the cell, and then you just have a slight rim of light purple around it. That helps you identify the agranulocyte or the lymphocyte. There's two kinds, the B cells and the T cells. B cells mature in the bone marrow, and they take care of like blood infection. T cells mature in the teeth, in the thymus gland, and take care of tissue infections. We'll talk a lot about these in chapter 12. You also have a cell called a natural killer cell. So they kill your cells that are infected with virus. So if you got COVID, these cells are trying to kill off the cells that the COVID is living in inside of your respiratory system. Okay, which is why we have so much damage to our lungs if you get that COVID infection, because they're killing your cells. Okay. Questions on this one? B cells, bone marrow, T cells, thymus gland, B cells, blood, T cells, tissue. And our last one, the monocyte, is the largest of all your red blood cells. Really, really big, has a nucleus that looks like a kidney bean. It's clear on the outside. You can also say it looks like a horseshoe, either way. So these are active phagocytes that leak out of your blood supply, get out into the tissues, where they enlarge and they become macrophages. Macro big, phage, phagocyte. So they're big eaters. They have an insatiable appetite and they eat up all the bacteria in sight. So they are more present in long-term infections. So if you're you're sick for a short period of time, that's the neutrophil. If you're sick for a long period of time, like I have chronic bronchitis, comes back all the time. So monocytes are going to be more active at that time. So long-term cleanup, they increase in numbers during chronic infections. Chronic means it's long-lasting or repeated. Acute means it's short-term and maybe like an individual event. So what they do, they take part in your immune system in that they present pieces of the bacterium or the virus that is invading your body. So they engulf it. Here, they're engulfing it. They digest it. So notice it's being broken down. Then little pieces get stuck to the outside of the cell. And then they show the outside. They're like, look at this protein that's in here. It doesn't belong. And they show it to the B cells and the T cells. The B cells will make antibodies to fight against it. The T cells will destroy the tissue that's infected. That's all part of chapter 12. Okay, so monocytes, they're the biggest, and they're in long-term infections. Platelets, that's your thrombocyte. Platelets. I think I gave you the word thrombocyte in the notebook. So platelets are what you use to clot blood. People who have hemophilia, they're probably lacking one of those blood proteins. It's probably not the platelets they're lacking, but I told you plasma proteins, there's fibrinogen. So there's like 12 different factors, proteins that are involved with the blood clotting process. They're most likely missing one of those factors, like factor 8. They have numbers. So these are needed for normal clotting of blood. When I had my hamstring attachment, I told you I ripped the hamstring off of my ischium, and they had to reattach it, and they screwed it in. So they drew blood out at the same time. They spun it in the centrifuge. They took out my platelets. They created a platelet paste. And then on the screw, then they put this platelet paste over the screw so that my blood would immediately start clotting to speed up the healing process. So this controls blood loss at the site of injury. So they're little fragments of cells. They're not full. I don't have a good picture. They're not full cells. So these little fragments stick together, and that's what creates basically your scab, right? It creates a mesh that stops blood from going through. That is what they are. We'll talk more about them later. Okay, last one. So here's our second intermission. Check for understanding. Raleigh can probably tell us what slide this is on because it's the one I was saying earlier. I got 5 minutes. See if we need more time than that. Miss Hefty's going to monitor, see that everybody is working, and she'll send you a chat saying, hey, dude, get on it. And double time. When we go into hybrid, Miss Hefty might be monitoring the virtual group while I'm monitoring the live group. We'll all be in the same place, but she might be talking to that group a little bit more, and I'll be talking to this group a little bit more. So a few minutes, what are the two categories? Not types of individual cells, but categories. They fit into one or the other. What are the two categories? Things I'll ask you on a quiz. Which one's most numerous? Which one's least numerous? Which one's biggest? This is part of what they do. Which one fights parasites? Which one is filled with histamine? One of your blood categories is lymphocytes. What are the two types of lymphocytes? And what is the function of platelets?